Our next speaker is Ozioma Okonkwo. I hope I got that right, Ozioma, from University of Wisconsin-Madison.
Thanks, everyone. It's a pleasure to be here this morning. When I first got the email from Molly Waxter about speaking at this summit, my first reaction was that this has to be spam email. Because how could I possibly have anything meaningful to say to such an illustrious crowd? But here we are. So this morning, I'm going to be speaking to you about some of the work that my colleagues and I have been doing in Wisconsin in the area of resilience to Alzheimer's disease. So as is very well known to all of us sitting here in this crowd this morning, age and ApoE4 carriage are the two most potent drivers of the pathologies of Alzheimer's disease in older age. And I think that the case for age was made in a very compelling manner in this autopsy series from the Braggs in 1997, where they demonstrated that the two core lesions for AD, that is the senile plaques and the neurofibrillary tangles, show a very clear age gradient, such that the more severe occurrence of these lesions are exclusively seen in older age. With respect to ApoE4, on the other hand, I think that one of the initial studies in this area, studied from the Duke group led by Alan Roses, again clearly showed that there is a marked effect of ApoE4 carriage on the risk for late onset AD. Here they show that depending on whether you carry 0, 1, or 2 copies of the ApoE4 allele, your risk for AD was accordingly increased. For example, looking at what happens at age 75, the percent of cases that are affected by this disease at age 75. What you see is that individuals who are non-carriers of the ApoE4 allele, there is only 24% of them have transitioned to dementia at that age. Then when you look at those who are heterozygous for ApoE4 carriage, that number jumps to 61%. And then when you look at those who are homozygotes, that's 86%. And in fact, in this series of 42 families that they studied, by age 80, essentially all the individuals had converted to AD dementia. So again, this couple of studies, drive home the point that age and ApoE4 really underpin risk for Alzheimer's. So against the backdrop of this information, then, my colleagues and I, our conceptual model for resilience posits that with the accumulation of risk, and in this case, risk can be thought from the point of view of aging or carrying 1 or more copies of the ApoE4 allele or other indicators of risk. Again, with the accumulation of risk, there comes to be a separation in the biomarker profiles of individuals such that you begin to observe a distinction in the phenotypic traits for AD, such that individuals who are low on this construct, which is yet to be defined, resilience, hit a hypothetical abnormality threshold at an earlier point in time compared to individuals who are higher on this construct called resilience. So to test this in an experimental fashion, then, or with scientific approach, we've been looking from the lens of physical activity and also cardiorespiratory fitness. And one of the initial studies that we did in the Wisconsin Registry for Alzheimer's Prevention with my student Stephanie Schultz, who is now working with Tammy and Ann Fagan at WashU, we asked the question, does engagement in physical activity attenuate the influence, the clearly known influence of age in the alteration of markers for Alzheimer's? And we used a multi-modal imaging approach to examine different aspects of the pathologies of Alzheimer's. But the first thing that we did was, again, to demonstrate in our cohort that truly, in this case, for this first study, age really is the biggest driver of these changes. And that was indeed what we saw, that when you look across each of these markers here, we found that there is a clear and distinctive effect of age or aging on all of these indicators for Alzheimer's. There was an effect for ApoE4, but that was only for amyloid burden. And again, in this series that we examined, we did not see an effect for family history of Alzheimer's, though, of course, we had published previously and since then on the effect of family history of Alzheimer's on risk for the disease. So again, this was just a first pass to demonstrate that it made sense to look at age when trying to address this question of resilience. And so going past that then, our models then showed that when you looked at amyloid burden, you see that when you examine those, so the red line, again, is those who are physically inactive, as determined by a failure to meet American Heart Association guidelines for moderate intensity exercise in older adults. Then when you look at those who are physically inactive, you see what you might expect, that there is this upward tick in amyloid burden in the brain with increasing age. Whereas if you look at those who are physically active, you do not see a corresponding uptake in amyloid burden in the brain. And of course, you see that there is a significant interaction there. And the simple mean effects show that indeed there is a significant impact of age or aging on amyloid among those who are inactive, but this is absent among those who are active. When we looked at glucose metabolism with FDG PET scan, the same is also true, that with, again, increasing age, there is a decrease in the uptake of glucose in the brain among those who are physically inactive, but not among those who are physically active. The same was also shown when you looked at hippocampal volume and episodic memory, that across all of these four modalities that were examined, there is an attenuation of the influence of age or aging on these markers for Alzheimer's as a function of level of physical activity. Now, a few years later, then we went back again to this finding here that ApoE4, as has been shown in several other studies, has a demonstrable effect on amyloid in the brain. And so we went back to look at that, but this point, and looking from the prism of CSF assays. And then now we computed a polygenic risk score that was based on 4 SNPs that are involved in cholesterol metabolism. And we see again that individuals who have higher scores on this polygenic score, that they have a reduction in CSF A-beta 42 compared to those who have lower genetic vulnerability. The same was also seen in the ratios of tau and phospho-tau to amyloid. Again, this is what one might expect. But then when we superimposed aerobic capacity on all of this, what we found, and this was consistent across board, is that influence of polygenic risk was only present among those who have reduced aerobic fitness. That when you look at those who have higher aerobic fitness, you do not see the same adverse influence of genetic vulnerability on the CSF markers for Alzheimer's. Shifting gears then a little bit from that, my foray into the potential genetic underpinnings of resilience came by as a result of a very chance meeting with this very brilliant scientist, Dina and Dubois from UCSF. We are part of the Paul Bissin Scholars Program funded by the NIA and AFARA. So we were just sitting at a table about 2 or so years back, and we got talking and realized that our interests actually were kind of similar. Now she is a basic scientist, and she had published a very seminal paper earlier that year, showing that the longevity SNP, Clotho, does enhance cognition in a very clear manner in knocking mice. And so I asked her if we could look at this in humans, and she said, why not? And so my original intent was actually just to kind of provide some resources for her to assay this SNP in the RAP cohort. But Dina, again, thanks to her junior, said, well, why not let's write a grant instead? And I said, sure. And and we did, and to my shock, the grant was funded the first time around, again, by Marley, thank you. And so we've since begun to look at the influence of this SNP in our cohort in Wisconsin. And our results so far suggest that this longevity SNP, that being heterozygote for Clotho, confers resilience in a very clear and marked way against the influences of age and ApoE on the phenotypic traits that characterize Alzheimer's. And we showed this with respect to again, CSF A-beta 42, that when you look at the individuals who are non-carriers of the resilient variant of Clotho, you see what you would expect, that individuals who are ApoE4 positive have a reduction in CSF A-beta 42 compared to those who are ApoE4 negative. But when you switch gears and look at those who carry the resilient variants, that is those who are heterozygotes for Clotho, you find that that same ApoE differential is absent. You really cannot tell if this person is ApoE4 positive or negative. They pretty much are at the same level. We also found the same when we looked at the effect of aging on phospho-tau and also aging on performance on challenging cognitive tests, that, again, among those who do not carry the resilient variants of Clotho, you see an aging effect, which is absent among those who carry the resilient variants for Clotho. So in summary then, we have become accustomed to thinking of age or aging and ApoE4 as non-modifiable risk factors for AD. But my hope is that the work that our group and other groups across the country is doing will lead us to begin to put a question mark after that statement. Maybe we cannot change age or aging per se, or cannot change one genetic makeup, but the data here suggests that we can influence the impact of those processes on one's risk for Alzheimer's. The next step then, I think, is to begin to think about mechanisms in a more rigorous way. How exactly does aerobic fitness or Clotho exert its impact on resilience? And I think that can only happen in the context of collaborations between basic scientists and clinical scientists, like the one that Dina and I have just established. And I think that if such collaborations are done well, they have the potential to lead to novel therapeutics for Alzheimer's disease and cognitive aging. These and similar other types of considerations are explored in more detail in a recent special issue that I had the good pleasure of working with Prashanti Vamuri, who is somewhere here, a very brilliant scientist, again, and a good friend. Thanks to Andy Sekin, the editor-in-chief, who allowed us to assemble this special issue on resilience and reserve, and to Jakob Stern and Gail Shettlard and Michael Evers, who are the leaders of the newly established professional interest area called Resilience and Reserve in AD. So Prashanti and I had the good fortune of co-editing this special issue in Brain Imaging and Behavior, where we assembled a host of scientists to address these notions of what is resilience, what's reserve, how do we measure it, how do they impact risks for Alzheimer's, and so forth. So with that, then, I would like to end here and thank the NIA for having so generously supported my work and the work that our group in Wisconsin are doing. I want to thank my boss, Sanjay Astana, who I think is somewhere there in the crowd, and my mentors, Sanjay. And also a special thanks to Virginia Wadley, who was my graduate school mentor. She and Dan Marson were the ones who really got me fired up about Alzheimer's and gave me the initial start, which has brought me and my lab to where we are today. Thanks, everyone.
So are you going to tell me that I don't need to exercise anymore if I've got am I heterozygous for clotho, or does it bump you even further up to give you more resilience?
That's an interesting question, and that actually is one of the aims of the R21 that Dina and I wrote, to look at the interaction between physical activity or aerobic fitness and clotho. One of the things that actually has been shown by some prior animal work is that physical activity elevates the level of serum clotho in the system. And so there seems to be potential there for some form of interaction between these two factors, but exactly how that instantiates is what we hope to tease out in this R21. Thank you.
A nice data and talk. So I have two quick questions. The first one, the interactive effect of the closal polymorphism with ApoE, is there any effect, relation with the gender effect of this? E4 females usually have high risk. Second question is the fact of the closal polymorphism on E4 effect. Have you guys did any correlation studies? Is it mediated by any A-beta effect, tau effect, any possibility there?
So I think the first question asked about the potential for the modification of ApoE4 effects on CSF amyloid by clotho to also have a gender differential. Mm-hmm. So far, we haven't found that to be the case, but again, this work has just been funded, and so we are at the very initial stages of exploring this. And I think I'm not sure I fully got the second question. Could you please repeat?
The second question is the closal effect, interact effect with E4, in the presence of E4, is mediated by any AD-related markers or just a direct effect?
So what we have shown is that clotho does not have a direct influence on AD markers, but it modifies the effect of ApoE on these markers. In fact, if I go back to this study here by, if I can get to there, this study here by Dina, in her animal work, she clearly showed that clotho did not influence the levels of plaques and tangles in the brain. So these knocking mice who were bred for clotho had about the same amount of plaques and tangles in the brain. However, their cognition did not suffer as a result. And I think that is the true meaning of resilience or reserve.
Okay, Singh.
I was trying to think about the mechanism that accounts for why the exercise is so protective. And so it struck me that CSF A-beta is a marker not only for brain amyloid, but vascular amyloid also. And so perhaps is it possible that by exercise, you're activating those pathways and therefore increasing the vascular strength, if you will, and therefore reducing the likelihood of blood-brain barrier breakdown, which would then mitigate the degenerative process?
Absolutely. That seems to correspond with our emergent findings in this area, that one of the key pathways through which physical activity or exercise influences risks for Alzheimer's is modification of the vascular pathways. So, excellent point.
Thank you very much. Thank you. Thank you very much for the very interesting talk. I was very intrigued by your statement that you said age is one of the most, the strongest risk factors for Alzheimer's. And my question is really we all age, so it's not like we all get dementia. So to consider as a risk factor, there's no difference. There's got to be a difference between the way we age that affects us for the risk. It's not just pure age. The number is a risk factor, right? And it's great to see that you're actually looking at lifestyle factors that are influencing this process. And I think that's probably the ultimate key to understanding why aging, why do you get this disease more with aging and not so much, when you're younger? And it's not any different than, other chronic diseases like heart disease, where you do have a higher risk for it when you get older. So my question is really also, should we be considering age as a risk factor, or should we really be considering the lifestyle factors that are affecting aging? And do you have any have you looked at other lifestyle components besides just exercise, like diet and sleep?
And
per
if
se?
you could answer that in 3 seconds.
Yeah.
I cannot agree more that clearly not everyone gets Alzheimer's with older age, but there also is no denying the fact that increased age is a clear risk factor for Alzheimer's. No one really gets Alzheimer's at age 2 or age 3. As far as other other lifestyle factors that might have an influence on this process, some of my colleagues have also been looking at things to do with diet and sleep, and trying to understand how those might modulate the influence of the aging process on risk for Alzheimer's. Thank you.
So, Ozioma, if I could ask you to hold that question for the general discussion so we can move on, please.